this imago stage. A holometabolous metamorphosis is when there's a distinct pupal stage between the larvae and the adult. And in this case, the larval tissues transform completely into adult tissues, and those tissues can be degraded completely, like in a butterfly, transforming from a caterpillar into a butterfly. There's almost complete histolysis, which is amazing that the body just breaks down into this goo, but somehow the memories that are held by the larvae, by the caterpillar, persist into the adult stage. So it's a pretty amazing transformation process. So the pupal cuticle then becomes a mold for the adult form in a holometabolous organism. And there's a lot of energy required, so a lot of fat has to be stored in the larval stages to allow for this complete metamorphosis. One of the differences, this was a question that was asked, is what are the differences between hemimetabolous and holometabolous adults? And one of the biggest differences is that holometabolous adults don't have compound eyes, but hemimetabolous adults do. So it's kind of interesting because they're going through a more complete metamorphosis, but they don't develop as complex a visual system. So here's a picture of some pupae. So exarate pupae have appendages that are free from the body. These are some caddisfly. There's actually a larva that's kind of heading into the pupal stage on the left of this diagram, and the pupa on the right. You can see the legs have developed and there's like some space for wings in there. Obtect pupae have appendages that are really glued down, so they can't move their legs and at all. Their appendages just don't move. They're more like little mummies. Some pupae can move when necessary, but their movements are very limited. So they can kind of wriggle or shiver. They can move a little bit, but they can't really like get away. So the pupal stage is often quite prone to predation. Some larvae will crawl out and pupate on land. So some beetles, megalopterans, and neuropterans. And some pupae attach to substrates, like was shown here in these rock cases for trichopterans. And then some pupae will burrow down into sediments or build cases for their final instar stage. So here's just some cool pictures of some of the different life stages. So on the left, 1, 2, 3, we see caddisfly larva, caddisfly pupa, and a caddisfly adult. And on the right, A, B, C, D, we see at the bottom, we see the larvae of a simuliid fly. Or sorry, we see the larvae is B. This is really silly. So the larvae is B. C and D show the pupa, both in the case in C and out of the case in D. And then A is the adult. So sorry about the order there. So pupae are very busy metabolically. So they're doing a lot of work. They're not really growing, but they're developing intensely. And so they need to exchange gases. And so they often have these respiratory horns at the surface or respiratory organs to somehow bring, especially if they're in some kind of a case, they need to bring oxygen into their case. Some of them might have to move up to the surface to get oxygen. Some might actually embed their respiratory horns into a plant like a macrophyte to access air inside the plant through its parenchyma. And many pupae, because they can't move very well, are vulnerable to predation. And so in particular, chironomids will prey on caddisfly pupa because they can't move. Okay, so eclosion is when the ferrate adult breaks out of its pupal cocoon, and then it starts to extend its wings, its limbs, and its antenna. So trichopterans tend to pupate underwater, and then the ferrate adult swims to the water surface using what's called phototaxis, and that just means it's sensing light and moving toward the light. And then eclosion, kind of the extension of the wings, occurs at the surface. Usually for aquatic insects, there's a transition from aquatic to terrestrial habitat. And eclosion is often really fast because they need to move through that ferrate stage to the point where they can fly away from predators. And so eclosion happens quickly. It often happens at night to protect the organisms from predators. And sometimes eclosion happens differentially. There's sexual dimorphism in eclosion. And if females emerge first, it's called protandry, and if females emerge first, it's called protogyny. And I already mentioned that the mayfly subimagos emerge underwater in their hydrofuge, which allows them to reach the surface. There's a variety of environmental influences on development. So temperature, photoperiod, humidity, water chemistry, oxygen availability, food availability, inter and intra-specific competition and predation can all influence development. In general, insects are poikilothermic ectotherms, which means that their body temperature mimics the environmental temperature. And so this means that increased environmental temperatures can increase development and metabolism. But typically, if there's increased temperatures, that spurs development and metabolism. What that means is that the final adult body size will generally be smaller than an organism that's developing under cooler temperatures. So there's some trade-offs there. We can use what are called degree days to measure cumulative temperature through time. And so instead of just looking at the number of days that it takes for an organism to develop, we can look at the number of days times the average temperature each day, and that gives us the degree days. And so if there's a whole bunch of days, like 10 days, but they're all cold, that would have a lower number of degree days than 10 days that were all really warm. Degree days are useful when measuring anything that's metabolic or influenced by temperature. And so we can also use degree days to look at changes in terms of like mass loss of leaves through time. So we often use degree days for decomposition studies. And then there's a bunch of terms for how many generations occur per year. So if you're a univoltine organism, you might only reproduce once per year. A bivoltine organism would reproduce twice. If you're multivoltine, you might reproduce more than twice. And if you're semivoltine, then you reproduce fewer than one time per year. Okay, so in terms of development, it can be stalled for a number of reasons. So dormancy is a state of suppressed development, and we're going to talk about two different types of dormancy, diapause and quiescence. So quiescence is an immediate response to some decline in a limiting variable. So, you know, like it gets really, really cold, or there's the stream dries up. This results in reduced activity and slows physiological responses. But the organism, if they're just experiencing quiescence, can bounce back quickly. So say it warms back up or water enters the system again, they can respond more quickly to that change, and they can exit this quiescent stage and increase metabolic activity. Diapause is a little bit more complicated. It's a genetically regulated process that interrupts development, and it can last for several months. And so it might be kind of pre-programmed in for an organism to go into a diapause period based on the history of environmental factors at a site. So when in diapause, insects can resist environmental stressors. So if a site is known for drying up every year, there might be this genetically predetermined kind of state of diapause that the organisms go in to resist that dry down period. Okay, so some sections of the population might enter this pre-programmed diapause timing, and other parts of the population might not. And so this allows for what's called cohort splitting. It increases genetic variation. It allows the slowdown of maturity and development for some parts of the population. And it kind of just spreads the wealth a little bit. It allows for diversity that allows populations to persist through environmental stresses. And then eggs can be in diapause for a very long time. And so they can kind of be seen as a seed bank for aquatic systems. Eggs can kind of enter diapause and hang out for several years in mud banks, in dried up lakes and ponds. And then when rains come back, they can hatch. So eggs are often in a diapause state for a while. And that again helps kind of just spread out the reproductive effort for a population. So what's a life cycle versus life history? These terms are used pretty interchangeably, but they're kind of different. So the life cycle is a qualitative description of the sequence of life stages. So egg to larvae to pupa, or in this case, egg to larvae to subimago to imago for a mayfly. That would be its life cycle. The life history of an organism is a little bit more detailed and involves both quantitative and qualitative details of events associated with the life cycle, and this can vary a lot between individuals or populations. So for example, you might have an individual that's laid as an egg, and then the egg goes through diapause. Maybe that leads to asynchronous egg hatching for the population. And so then you end up with a population spread among multiple instars. They're not all developing at the same rate. And then you have different hatching timings because of those differences in instar development. And then you might have the organism, once it's an adult, it's a bivoltine mayfly, or this part of the population is bivoltine. It might undergo facultative parthenogenesis, and then it might have high fecundity. So see, these are all different kind of aspects of the life history of an organism or a population. So it's just a little bit more detailed and goes into what actually happened at each of these life stages for this individual. And that's the end. So I hope you enjoyed it, and we'll see you next week.